Okay, today I want to explain about laboratory equipment, autoclave and magnetic stirrer. Okay, the function of autoclave. An autoclave is a machine that provides a physical method of sterilization by killing bacteria, virus, and even spores present in the material put inside of the vessel using steam under pressure. This is a picture of autoclave machine. Where autoclave commonly used? Where we can find it. So commonly autoclave are used in healthy care setting to sterilize medical device. In medical labs, autoclave are used to sterilize medical equipment, glassware, surgical equipment, and medical waste. The next slide is technical feature of autoclave. The autoclave series has a chamber volume range from 120 liters to 310 liters. The autoclave operates with saturated steam as the sterilizing agent with a temperature range from 105 Celsius to 138 Celsius and a working pressure that meets ASME and PED requirement. What is ASME? SME is American Society of Mechanical Engineering and PED Pressure Equipment Directive. Daily plan preventive maintenance for autoclave. First, make sure the chamber drain strainer located in the front drain hole of the sterilizer is totally free of debris. A clogged chamber drain strainer will prevent the sterilizer from sensing temperature. 2. Be sure to immediately wipe up any spill in the autoclave chamber with chamber cleaning solution and accompanying cleaning pads. Do not use bleach containing hypochlorite, corrosive chemical, acid, or seawater inside the stainless chambers. 3. If your autoclave is equipped with a steam generator, it may have an automatic generator blow-down valve on it. If the generator exhausts the steam each day, when you turn it off, then it has an automatic blow-down. If it does not, you should manually drain the generator at the end of each day, depending on water quality. This will aid in drawing off the sediments and impurities in the water and keep the generator clean. Next, plan preventive maintenance for autoclave weekly. First, you must flush the chamber with clean, non-chlorinated water or chamber cleaning solution. This is particularly necessary if saline solutions are being sterilized. Depending on the type of goods being sterilized, the chamber may also require scrub cleaning or other type on a regular basis. 2. Inspect the door gasket for tearing, pitting, or warping. If you see any of these, it may be time to replace it. 3. Visually check for water and oil stream leaks. If you see a leak, find the root cause and repair it as soon as possible. Common issue with autoclave. The problem: the sterilizer won't reach the temperature set point, so you must check and clean the clog in the chamber. That is preventing air from escaping the chamber and the steam from reaching the correct pressure and temperature. 2. The lot is damaged by the autoclave, so you must run a specialized liquid cycle that releases the chamber pressure slowly instead of all at once. This gives the liquid time to cool down, so it isn't at its boiling point when the pressure is released. All consolidated autoclaves are equipped with a liquid cycle. 2. Magnetic stirrer function. A magnetic stirrer ensures proper mixing that can boost sensor response time, allowing measuring instruments to achieve stable reading more quickly. Where magnetic stirrer commonly used? So, we can see magnetic stirrer are commonly used in biological lab and microbiology lab to cause a stir bar absorbed in a liquid to spin quickly. Biological lab and microbiological lab make use of magnetic stirrer as it is small in size and easy to move it from one place to another. Even the device is easily cleaned and sterilized than any other laboratory equipment. Technical feature of magnetic stirrer. Basically, the magnetic stirrer must have a maximum steering speed 100 to 1000 rpm with stable speed control and good speed stability. Heating capacity must 600 watt and hot plate size basically 175 millimeter. The supply must 220 to 240 volt and 50 Hz. The hot plate should be chemically resistant to acid and alkali. The top plate material must stainless steel. Steering unit should be enclosed so that corrosive fumes do not enter it. Control for both hot plate and stirrer should be provided with suitable indicator. Necessary electrical cable should be provided. Plan preventive maintenance for magnetic stirrer. First point, always use fresh buffer for calibration. Choose buffer that are no more than 3 pH unit apart and bracket expected sample pH. Check electrode slope daily by performing a 2-buffer calibration. Slope should be 92 to 102%. Between measurement, rinse electrode with distilled water. Stir oil buffer and sample during measurement. Place a piece of insulating material, styrofoam cardboard, between magnetic stirrer and beaker to prevent error from transfer of heat to sample. Common issue with magnetic stirrer. The problem is uncoupling of the bar becoming stuck, insufficient mixing. So, you must increase the speed slowly. Use the correct stir bar. 3. Try switching vessel. 4. Use a unit with more power. And 5. Consider using an overhead stirrer.
Hi, Assalamualaikum. My name is Sabrina Firdaus, and today I'll be talking about biological microscope and automated blood coagulator analyzer. This is a biological microscope, and this equipment is under laboratory class of medical device. As you know, there's 6 class of it: diagnostic, therapeutic, laboratory, radiology or imaging, rehabilitation, and palliative care. Microscope is used to study organisms and their vital processes. It is also used in science laboratories to visualize every minute objects such as cells, microorganisms, giving a contrasting image that is magnified. Here you can see every piece and little details about microscope and also the name of it, so I'll be explaining what its function. Eyepiece is on the top of it. It's also called as ocular lens. This is the part used to look through the microscope. It's found at the top of the microscope. Its standard magnification is 10 times with an optional eyepiece having magnifications from 5 times to 30 times. Eyepiece tube is the eyepiece holder. It carries the eyepiece just above the objective lens. In some microscopes, such as the binoculars, the eyepiece tube is very flexible and can be rotated for the maximum visualization for variance in distance. For monocular microscope, there are none that flexible. Objective lens is the three lenses here. These are the major lenses used for specimen visualization. They have a magnification power and that um, of 40 times to 100 times. Let me show you. This is the objective lens. The nose piece is in here, also known as the revolving turret. It holds the objective lenses. It is movable, hence it can revolve the objective lenses depending on the magnification power of the lens. The adjustment knobs is the knobs that are used to focus the microscope, probably here. And there are two types of adjustment knobs. It's actually adjustment knobs and the coarse adjustment knobs. This is the coarse adjustment knobs. The stage. Here's the stage. This is a section where the specimen is placed for viewing. They have stage clips which hold the specimen slides in place, and the most common stage is a mechanical stage, which allows the control of the slide by moving the slides using the mechanical knobs on the stage instead of moving it manually. This is the mechanical stage, by the way. And the aperture here, this is small things. This is the hole on the microscope stage through which the transmitted light from the source reaches the stage. Microscopic illuminator. Here it is. This is the microscope light source, and it's located at the base so that it is used instead of a mirror. It captures light from an external source of a low voltage of about 100 voltage. A condenser is here. As you can see, these are the lenses that are used to collect and focus light on from the illuminator into the specimen. They are found under the stage next to the diaphragm here of the microscope. They play a major role ensuring clear, sharp images are produced with a high magnification of 400 times and above. The higher the magnification of the condenser, the more the image clarity. More sophisticated microscopes come with an app condenser that has a high magnification of about 1000 times. The diaphragm, the most infamous part, it is also known as the iris. It's found under the stage of microscope, and its primary role is to control the amount of light that reaches the specimen. It's an adjustable it is an adjustable apparatus, hence controlling the light intensity and the size of the beam of light that gets into the specimen. For high quality microscopes, the diaphragm comes attached with an app condenser, and combined that they are able to control the light focus and light intensity that reaches the specimen. Condenser focus knob. Focus knob. This is a knob that moves the condenser up and down, thus controlling the focus of light on the specimen. App condenser. Well, we've been talking about app condenser just now, from condenser to diaphragm, and this is actually the app condenser. It is actually especially designed on high quality microscopes, which makes the condenser to be movable and allows very high magnification of above 400 times. The high quality microscopes normally have high numerical aperture than that of the objective lenses, so it's very different. It varies, and the function is also different. The rack stop. The rack stop is it controls how far the stages should go, preventing the objective lens from getting too close to the specimen. And if the lens is very close to the specimen, it may have damage on the specimen itself. It is responsible for preventing the specimen slide from coming too far up and hit the objective lens. Here's the ppm list. This is the microscope daily cleaning checklist from the Canton City Health District. And of course, it's different for every device, different for every places or the function of itself. For this one, it only have four. So it's just turn off light, use lens cleansing fluid on lens tissue to wipe the lenses of dust, debris, or oil. If body fluids are suspected, contact the scope, use bactericidal disinfectant to decontaminate and replace dust cover. This is very important actually, because it actually helps to prevent any microorganism gets into the person who are in charge of the microscope, and prevent that device are always ready. This is actually troubleshooting in the most common problem. There are two, so focusing area and device won't turn on. The first one is focusing area. The height of the microscope condenser might be set too high or too low. This can also affect your microscope resolution. Make sure your microscope objective lenses are screwed into the body of the microscope all the way, and your microscope cover slip may be too thick. So you might check on these three first, and then you can troubleshoot it. The device won't turn on. If you happen to have this problem, you can check to see if your microscope is plugged in, check to see if the bulbs are installed correctly. You may have to install or reinstall the microscope bulb. If the bulbs are installed, check to make sure they are not loose, which sometimes happen during the shipment. And check the rail stud, which is the light intensity control, on the side of the microscope. Check the fuse. Is the fuse is good, or all microscope with corbs have fuses that can be accessed from the outside of the microscope. The second device we'll be talking is automated blood coagulator analyzer. This device is also on the laboratory medical device. Its function is for measure the clotting mechanism of hemostasis, used primarily to detect clotting deficiencies related to thromboembolic disease, thrombocytopenia, impaired liver function, hemophilia, von Willebrand disease, and other. The number of device. KC4 is not connected to power supply, or power supply is not connected to the outlet. What you can do is assure that the connecting cable is seated firmly in the power supply socket. Assure that the power supply cord is connected to the appropriate outlet. Number 2 is after pressing on and off, the temperature fails to stabilize at 37 Celsius. Measurement well is rotating. This is an instrument error, which non-functional sensor or thermostat is overheated. What you can do for the corrective action is place approximately 3 ml of water into a 15 mm of test tube. Insert a thermometer, observe the temperature after 10 to 15 minute equilibration period. Number 3, the last one, is controls are in range, unexpected results on patient samples. Now, a lot of samples we can expect it to be some sort of result, but if it's far from what we expected, then we can probably blame it on a device. So there's two. It's pre-analytical error, whether it's overfill or underfill of collection tube, or incorrect value, such as the type of EDTA or heparin, and the concentration or lack of anticoagulant. What you can do is full draw in commercial vacuum tubes assures correct blood or anticoagulant ratio, or use anticoagulant as recommended by the region manufacturer. This is the ppm checklist, which very clear. It's just for cleaning. Clean the rack area thoroughly. Check and clean the pipettes used in the device. Make sure region warming wells and reaction incubation wells also has been wiped clean, and the screen should be daily cleaned to make sure data received are clear. This is very important for make sure the device is always on the go and the people can. So that's it for two device. Thank you.
The next equipment that we're going to be talking about is the autohematology analyzer and the blood gas analyzer. Autohematology analyzer. Function of the equipment. Hematology analyzers are used to count and identify blood cells at high speed and accuracy. They are also used to conduct a complete blood count, which is usually the first test requested by physicians to determine a patient's general health status. A complete blood count includes red blood cell, white blood cell, hemoglobin, platelet counts, as well as hematocrit levels. This equipment is commonly found in a pathology laboratory. Pathology is a significant field in modern medical diagnosis and medical research. The features of this device is 1. It is fully automated, compact, and cost-effective. Second, it is a 3-part differentiation of WBC. Third, it can test up to 30 samples per hour. And 4, it can also store up to 10,000 sample results. 5, it has 3 counting modes. 6, it came with a built-in thermal recorder with optional external printer. And last but not least, 7 is it has a really large color LCD display. Preventive maintenance. Here in this picture attached is an example of preventive maintenance for this equipment. Next is troubleshooting. If the sample is sufficiently mixed, what we're going to have to do is remix the sample, analyze the sample twice more. If the two results are equivalent but different from the problem results, we draw and rerun the sample. Then, if your problem is there a missing bubble problem, make sure to check the valves in the mixing bubble tubing are working correctly. If they're not, replace problem tubing with a new one. The third common problem, is sample only partially aspirated. What you have to do is to rerun the sample, hold the vial against the front of the aspirate probe until you hear a beep. Blood gas analyzer. The function of the equipment, a blood gas analyzer, analysis tests blood to measure blood gas tension values. It also measures blood pH and the level and base excess of bicarbonate. The source of the blood is reflected in the name of each test. For example, arterial blood gases come from arteries, venous blood gases come from veins, and capillary blood gases come from capillaries. Clinical significance of this equipment. Blood gas tests can be used in diagnosis of a number of acidosis conditions, such as lactic, metabolic, and respiratory acidosis, diabetic ketoacidosis, and also of respiratory alkalosis. Particularly, umbilical cord blood gas analysis can give an indication of preceding fetal hypoxic stress in combination with other clinical information. Normal paired arterial and venous cord blood gas results can usually provide a robust defense against the suggestions that an infant had an interpartum hypoxic ischemic event. Abnormal results may be due to a wide range of diseases, including poisoning and trauma, as well as lung, kidney, or metabolic diseases. Drug overdose and uncontrolled diabetes may be determined from abnormal results. Head or neck injuries or other injuries that affect breathing can also lead to abnormal results. This equipment is also commonly found in a pathology laboratory like autohematology analyzer. Both of the equipment is commonly found in a pathology laboratory. These are the features of this equipment. These are the technical features of the blood gas analyzer. Preventive maintenance. Troubleshooting. If your problem is multiple PT failures on one or more analytes, what you have to do is to check whether the method code appropriate for analyzer, or you can also check your barometric pressure data whether it is entered correctly or not. Then, if your problem is one or more PT failures occurring with instrument flags, what you have to do is you have to do a cleaning or maintenance of the instrument is required.
The urine analyzer is a device used in the clinical setting to perform automated urine testing. The units can detect and quantify a number of analytes, including bilirubin, protein, glucose, and red blood cells. Many models contain urine strip readers, a type of reflectant pottometer that can process several hundred strips per hour. This is urine analyzer component. From the front, we have printer lid, screen, keyboard, and feed table. And the back, we have serial port, two USB port, electrical power outlet, fuse, and power switch. is urine analyzer technical feature. These are the troubleshoot of urine analyzer. First is missing critical target. Second is availability problem. Third is a pre-pass. Fourth is limited control mode. Five is non-cellular material. And six is preparation time. The electrolyte analyzer is a device for measuring the electrolytes in the human body. They are primarily used in the quantitative measurement of sodium, potassium, and chloride in whole body, serum, or plasma. These are a few components of electrolyte analyzer. First, we have electrode module. Second, we have reagents. Third, we have peristaltic pump. Fourth, we have waste chamber. And five is sample probe. These are the technical features of electrolyte analyzer. This is the troubleshoot of electrolyte analyzer. First, the plug and seat loose motherboard. Second, the detector itself is broken. Third, fixing screws on the motor spool rotation axis is not securely in place. And fourth is the value cannot be rotated tightly. How to make sure your equipment lasts longer? You need to read the user guide. And second is know your machinery. Before using a piece of equipment, ensure you and all other operators know how it works and what is it for. Third is inspect regularly. Regularly inspection on equipment can point out inconsistencies and issues that may not be noticed in day-to-day use. 4. Carry out regular maintenance using a schedule. If something goes wrong, if it usually imperative that it is fixed quickly and correctly. Regular servicing can avoid unnecessary large outlays. 5. Replace parts when needed. With equipment that is being used a lot, smaller parts will wear and need replacing to keep it running to the best of its ability. 6. Clean after use. Cleaning equipment after using, especially equipment being used for concrete or mud and waste, will prevent pull of excess in moving parts, which can cause breakage or loss of movement. 7. Repair and refurbish rather than replace. When your equipment starts looking tired and doesn't require work as well as the day you bought it, it is tempting to go with the easy option. However, some suppliers will offer a repair and refurbishment service to get it back into top form. Store correctly. Storage is important when it comes to looking after equipment, especially smaller pieces of equipment that can easily get lost or damaged on a busy construction site. 9 is use quality equipment. As famously quoted by Warren Buffett, price is what you pay, value is what you get. As with anything in life, you get what you pay for. If you are willing to spend a little more for a superior quality piece of equipment, the likelihood is that you will be using it long after you would an interferopies.